Hello friends, welcome back. I am Professor Pranav Singh Patil and you are studying weed management. In last class we discussed about herbicides, the classification of herbicides, the chemicals like adjuvants and roll-up adjuvants in improving the performance of herbicides. Going to discuss another topic, that is mode of action of herbicides. Now we are going in depth, we are going to study the physiology of movement of herbicides in plants and how actually the plant gets killed. Okay. Not just the application, not just the how the herbicides can be used, but how actually the plant gets killed. So we are going in depth in the botany, in the physiology, and today we will discuss about the plant physiology of how the herbicides kill the plants. There are a number of herbicides available in the market. Different herbicides have different modes of action, and one by one we will discuss about how the herbicides actually kill the plants. So this is not a one event, it is a sequence of events from absorption of herbicide into the plant until the plant dies. Whatever the processes happen step by step, all processes together called as mode of action of herbicide. The sequence of events from absorption, how the absorption takes place, until the plant dies, how the plant dies, why the plant dies, all the steps are called as the mode of action of herbicides. So if I show you a picture here, suppose this is a weed and you are applying some herbicide. So herbicide comes in contact with the weeds, then it gets absorbed through the pores inside the leaves or through the cells from one cell to another cell, and then it moves inside the plant body. So movement will occur and the herbicide will go to the site of action, means it will go into some chemical reaction inside the plant and it will stop that important reaction and will kill the plant. So the all steps together is called as mode of action of herbicides. Together sequence of events are called as mode of action of herbicide. Why it is important to know the mode of action? If you know the mode of action, you know how the herbicide performs. Okay. All herbicides should not be sprayed by a single method. You should know how the herbicide actually performs, what it does. So then you can use that herbicide more efficiently. So it improves the herbicide performance. If you use the herbicide according to its mode of action, you can improve its performance. You can save the herbicide, you can save the cost, you can improve the performance. It also helps to diagnose the herbicide injury. By mistake, if you have applied a wrong herbicide, then looking at its symptoms, you can understand which herbicide has been sprayed by the farmer, and then you can do something about it. So that is diagnosing the herbicide injury if by chance if it happens. How do the herbicide kill the plants? That we want to know. So basically they inhibit a process in the plant that is essential to life. How the herbicide kill the plant? They go inside the plant, they are absorbed inside the plant and they stop certain essential process, and that is why the plant cannot complete its growth further and it will die. They inhibit a process in the plant that is essential to life. So what are the processes essential to life of a plant? Photosynthesis, respiration, protein synthesis, carbohydrate synthesis, lipid synthesis, wax synthesis, then amino acid synthesis, cell division, cell multiplication. What are the different processes? Chlorophyll synthesis. Number of processes are there and they are inhibited by the plant, and then the plant cannot grow further. Mode of actions of herbicide. So based on these processes, which process the herbicide stops and the mechanism of herbicide, mode of actions are designed. So according to different books there will be one or two extra and changes will be there, but these are the basic. Growth regulator mode of action. Number one is growth regulator mode of action. We will see one by one. Later on we will describe them. Amino acid inhibitors. Amino acid inhibition action are also known as protein inhibitors. Cell division inhibitors. So, the herbicide which stop the cell division. Cell membrane disruptors, the herbicide which destroy the already established cell membrane. Lipid biosynthesis inhibitors, pigment synthesis inhibitors, photosynthesis inhibitors, they will stop photosynthesis, inhibition of germination process. So, these are the some basic mode of actions, we will discuss one by one. Growth regulator herbicides or hormone herbicide, they are also known as hormones. Growth regulators. So, when these herbicides come in contact with the plant, they are responsible for sudden increase in growth of plant or sudden decrease in growth of uneven growth of cells will takes place. The plant will lose its natural growth and uneven additional growth takes place. That is the role of growth hormones. You know the growth hormones, plant growth hormones, auxins, cytokinins, gibberellins. So, those hormones can be used as herbicides. One such hormone was 2,4-D. People were using it as hormones. So, one farmer used it in large quantity, and then 2,4-D was known to world as herbicide. So, if you use it in ppm, it is a hormone, it is a auxin, but when you use it in kilograms, it becomes a herbicide, because it disrupts the normal natural growth and plants uneven growth takes place and it cannot survive and it will die. So, it is also known as hormone herbicide. So, these are the auxin type of, most of them manufactured are auxin type. They interfere metabolism of nucleic acid. They do not allow the normal mechanism of nucleic acid, and uncontrolled growth of cells takes place. Uncontrolled growth of cells takes place. There is a collapse of, that is why there is a collapse of symplastic and apoplastic systems. Symplastic, the living bodies, in living parts in the cells, apoplastic, the non-living parts in the cells. Because of the cell growth, abundant cell growth, the symplast and apoplast will collapse, that affect the phloem and xylem transport, and that is why the plant dies. The roots and stems consume the most of the photosynthesis. Leaves and secondary roots are starved. Because of the uneven growth, the roots and shoots consume maximum food, whereas the secondary leaves and stems do not get any food and the plants die. So, this is the what actually happens I have described, but the mode of action is sequence of events. So, what is the sequence of event that we will discuss? First of all, I will show you the symptoms. Uneven growth of cells. Can you see the uneven growth of cells? As if it is an attack of virus, just like attack of virus, uneven growth of cells, and how the mode of action takes place. So, most of them are the foliar applied. When you apply on the foliage, these herbicides are then absorbed by the plants and they are radially transferred in the body. So, they are translocated from one part to another part. So, if you spray on the leaves, they reach the roots also and they move inside the system. The site of action is the hormone receptors inside the cell. So, the hormone receptors inside the cell receive the herbicide. They affect the normal functioning of nucleic acid. Abrupt growth of cells takes place. Twisted, malformed leaves and stems develop. Twisting and malformed stems and leaves, and the examples are 2,4-D, dicamba, popular hormone type of herbicides. Second one is amino acid or protein inhibitors. We all know that amino acids are the building blocks of proteins. Proteins are developed from the amino acids. Proteins are essential for multiplication of cells, growth of plants. So, specific herbicides are there. Some herbicide bind to specific enzyme. Herbicide gets bind to specific enzyme and prevent development of essential amino acids. There are 20 essential amino acids and they are essential for development of proteins. But if amino acids are not developed, then protein cannot be developed. So, herbicide they get bind to specific enzymes the enzyme which is essential for production of amino acids, and amino acid cannot be produced because of lack of that enzyme. Which enzymes are there? Some essential enzymes are ALS, EPSP enzyme, glutamine enzyme. These are some examples. These enzymes are critical for production of amino acid, and their functioning is inhibited by the herbicides. ALS enzyme is an example of herbicide is imizethapyr pursuit. Acetolactate synthase is the enzyme ALS, whereas the EPSP enzyme is inhibited by glyphosate Roundup, enol pyruvyl shikimate phosphate synthase enzyme. The mode of action is amino acid inhibitors. Most of them are the foliage applied, some of them are the soil applied, but glyphosate is mostly foliage applied. So, they are readily move inside the plant, they are absorbed and move from root to shoot, or if you are applying on the soil, if you are applying on the foliage, it moves from leaves to stem and stem to roots all over the plant body. So, they are translocated and they inhibit the site of action, that is they inhibit the EPSP enzyme or glutamine enzyme or ALS enzyme. So, depending upon the herbicide, they inhibit some enzyme which is critical for production of amino acid. Plants results in stunting. Okay. First there is a stunting of growth, and slowly the plant gets starved and necrosis occurs and plant dies after few days. That is why the symptoms of glyphosate are not visible immediately on the same day. You need 2 to 3 days time for the glyphosate to show the symptoms. So, these are the symptoms. Slowly the necrosis of the leaves takes place, yellowing and the necrosis of leaves takes place. Inhibition of cell division, third. Some interfere the transport of precursors into the nucleus, which are essential for DNA synthesis. DNA synthesis is essential for cell division, and transport of precursors into the nucleus is affected by the herbicides. So, herbicide do not allow the precursor transport. That is why the cell division does not take place, DNA synthesis does not take place. They modify the chemical and physical properties of DNA. The herbicide modify the chemical and physical properties of DNA. They also inhibit the cell wall formation. So, which are these herbicides? Pendimethalin and butachlor. Pendimethalin storm, butachlor machit. These are the herbicides. How the mode of action takes place? The mode of action of the cell division disruptors, also known as seedling growth inhibitors, are most of them are applied on the soil surface. So, they are the pre-emergence herbicides or pre-plant herbicides. Up till now we have seen most of the post-emergence, now pre-emergence. So, they are applied on the soil surface. So, they remain. So, weeds when they germinate, they come in contact with these herbicides, and most of them are having limited movement inside the plant. So, they are contact herbicide, not translocated herbicide. They have limited movement inside the plant body, and they inhibit the cell division or they inhibit the mitosis process. So, that is why it causes stunting of the plant shoot and swollen root tips. If they are first absorbed by roots, then swollen root tips. Pendimethalin, butachlor are the examples. The injury to the crops can be seen, swollen root tips. You can see swollen root tips of some cereals when there is excess of this herbicide. Cell membrane disruptors. Cell membrane. So, already established plants are there. So, we are applying on the foliage of these plants or weeds, and we are disrupting the membranes. So, these herbicides, they form a toxic compounds like superoxides and hydroxyl radicals, which then destroy the cell membranes. The herbicides, when absorbed, they form toxic compounds like superoxides and hydroxyl radicals, which destroy the cell membranes. The oxidative degradation of lipids is responsible. Oxidative degradation of lipids takes place. Free radicals steal the electron from lipids. Free radicals steal electrons from lipids in the cell membranes and results in cell damage. These electrons are further they had to be transported for production of the other products, but these electrons are absorbed by the herbicides. Then the further process cannot take place. Paraquat and oxyfluorphane are the examples. So, cell membrane disruptors, they are, some of them are foliage only, some of them are both soil and foliage. So, they have limited activity, they are also contact type of herbicide. So, they react with the plant to form toxic compounds like superoxide and hydroxy radicals, and rapid necrosis of the stems and leaves takes place. They have very rapid reaction. So, if you apply the paraquat immediately on the same day, you will see the symptoms, rapid necrosis of leaves and stems. Paraquat, diquat, oxyfluorphane, these are some examples. Bleaching, you can see rapid bleaching, desiccation of the plants takes place. Lipid biosynthesis inhibitors, they prevent the production of fatty acid, the membranes cannot form. Quisalofop P, Targa super is one such example. They prevent the production of fatty acids, membrane cannot form, act only on annual and perennial grasses. So, this is a herbicide which is not a broad spectrum, it is narrow spectrum, they kill only grasses. Mode of action is the lipid biosynthesis, they are most of the foliage applied herbicides, they are applied on the foliage, so they are absorbed from the foliage and they are radially move inside the plant body. They are translocated type. So, applied on the leaves, they go up to root system. They inhibit the enzyme critical for production of lipids. They stop the enzyme which is critical for production of lipid. And stunting and death of tissue within the growing points of the plant takes place. Because of the lack of the production of lipid, there is a stunting and death of the tissues within the growing points. Diclofop, fluaziflop, these are examples of the lipid biosynthesis inhibitor. Quisalofop, so they mostly kill the grasses. Photosynthesis inhibitors, they block the electron transport chain of photosynthesis. So, there is a photosynthesis, photosystem 1, photosystem 2, and we know that there are a number of electron carriers, and finally, we produce the ATP and NADPH. So, these electron carriers, the electron transport system chain is blocked by herbicide by affecting its carriers. Hill reaction, that is the breaking of water molecule, is affected by herbicide. Herbicide inhibit the photolysis of water by deactivation of water splitting enzymes. They deactivate the water splitting enzymes. Photolysis of water cannot take place. Hill reaction cannot take place. Inhibition of respiration by uncoupling the oxidative phosphorylation. The oxidative phosphorylation process is disturbed by herbicide. Inhibition and inhibition of photosynthesis, both the examples we can see. So, photosynthesis inhibitors, they are mostly soil applied, okay, and they radially absorbed by the plant, radially move inside the plant, means they are translocated type, and they block the electron transport system of the photosynthesis, that is the mode of action, actual action, and the plants then become yellow and then become necrotic, okay, because of the lack of photosynthesis. Atrazine, simazine, metribuzin, the most common examples that we popularly use in cereal crops, they are the photosynthesis inhibitor type of herbicides. These are the symptoms of atrazine. Pigment inhibitors, the word itself suggests that they affect the key pigments essential for plant growth. The pigment essential for greenness of the plant, carotenoids, they destroy the green pigment, chlorophyll or carotenoids. That is why these herbicides are also known as bleaching herbicides. They cause bleaching of the plants, loss of color of the plants. The leaf color becomes white, bleaching, and most of them are soil applied, they are absorbed by the roots, clamazone, amitrol, These are some examples. So, pigment inhibitors, they are applied on the soil. So, they are pre-emergence or pre-plant type. They readily move inside the plant body. They inhibit the carotenoid biosynthesis. So, chlorophyll loss and foliage which lack color or bleached leaves. So, after application the foliage cannot produce the carotenoid, the chlorophyll production stops and plant dies. Amitrol, fluridone. Okay, you can see the bleaching of leaves of the applied herbicide mesitron. And finally, last one is the inhibition of germination. Some herbicide inhibit germination by stopping gibberellic acid production or inhibition of amylase production, which is essential for germination process. Alachlor and propachlor are some examples. They stop the gibberellic acid production, essential for germination of seeds. So, these are the mode of actions of herbicides. So, today we discussed in detail how individual herbicide actually step by step kills the weed, that is called as the mode of action of herbicides. Thank you. Today we will stop here.